Hey, everybody. My name is Keith Johnson. I'm the Purdue University Extension-Forage Specialist, and I will be your host today. Our hostess that's typically here, Alicia Rogers, unfortunately had some episodes of pretty extreme ice in northeastern Indiana where she lives, and they may be dealing with that on their farm or in the region yet. So I am pleased to step in on behalf of her and be your host for today. It's my pleasure to introduce our guest speaker. Dr. Grant Burcham is trained as a veterinarian and as an animal diagnostician, and he's part of the Purdue University Animal Disease Diagnostic Laboratory. And he is down at the Heeke laboratory, which is at the grounds of the Southern Indiana Purdue Agricultural Center in Northeast DuBois County. I've gotten to know Grant during his time as a diagnostician. He's an exceptional individual, and I know he's an exceptional diagnostician. And he's going to bring to you some of the things that he has seen there as a diagnostician at the laboratory, and some other considerations for us as we think about potential problems with the feedstuffs that we take to our livestock. So, Grant, thank you for being here. And if you want to share your screen, if you're going to do that, you may do so.
Thanks, Keith. Appreciate being asked to be here. We're going to visit a little bit about forage-related livestock diseases, and Alicia asked me to present. First of all, I hope everybody can hear me okay. And I'll try and monitor the chat. So if you want to put something in the chat about volume or anything like that, let me know. But Alicia asked me to visit a little bit about diseases associated with favorable forages. So those species that we intend to be forage species can have some issues, some toxic principles inherent to the forage or that occur secondary to fungal overgrowth in favorable forages. And then also diseases associated with stored forages. Now's the time of year I think a lot of people are using stored forages. We're going to talk specifically about preserved forages like haylage or baleage or silage. Let's see here. All right, so this is what we're going to cover first, a little bit about nitrate accumulating plants, the disease known as grass tetany, bloat, Dallas grass, some saframine issues, and then silicate urinary stones. So we'll start off with nitrate or nitrite accumulating plants. Some sources are listed there on the slide. Corn, Sudan grass, sorghum, and Johnson grass are probably the most common that we deal with. Those are the favorable forages that can accumulate nitrates. And accumulation can occur in different conditions, drought or high levels in the soil, for instance, if the soil has just been fertilized. Ensiling can reduce the content of nitrate, but baling and drying does not. If you're looking at drought-stressed corn, for instance, different parts of the plant will accumulate nitrates at different levels and different rates. And so if you look here, you're looking at kind of the bottom third of the corn stalk is the one where nitrate accumulation is going to be highest. And if, for example, you're grazing corn stalks, that's what's left out in the field oftentimes. So that's something that you want to be wary about, I think. How does this happen? How does this affect our livestock species? Well, if we're talking about ruminants, rumen bacteria normally transfer or transform nitrates into nitrites, ammonia, and then protein. But if the intake of nitrate is exceeding the capacity of the bacteria to reduce nitrate to nitrite and then nitrite to ammonia, we can have nitrate accumulation into the blood. And what that does is it messes with hemoglobin, and hemoglobin is the substance that binds oxygen in the blood. And so on the right-hand side, the tube that has the real red-looking blood, that's healthy. That's blood that has oxygen binding to hemoglobin. But when hemoglobin is transformed by nitrites into methemoglobin, it doesn't carry oxygen very well. And so it turns into that real chocolate brown color that you see on the left. That's one of the ways that we can recognize nitrate toxicity in animals, brown mucous membranes, and then clinical signs related to reduced oxygen carrying capacity in the blood. So animals will become incoordinated, they can't do very much. They may become weak. You may see some heavy breathing. That's the acute situation where animals are acutely affected by nitrites. After that, we need to consider situations where there may be subacute toxicity or chronic toxicity. And you may or may not see acute poisoning and then subacute poisoning later. You just, depending on the dose, may just see the subacute situation. Animals may just not look great, decreased milk production if you're doing dairy cows. And then one of the things that I've started looking for most recently, I test all of my aborted bovine fetuses for this, is you can see abortion and infertility in cows. How do we diagnose this? As Keith mentioned, I'm a diagnostician, so I'm really concerned. My day job is diagnosing disease in animals, so I'm really concerned about the diagnostic side of it. And if anybody has any questions relating to diagnostics, you can get a hold of me and we can figure out what samples to test and what can be done. But that's kind of what we do here at the lab. We can test forages or hay or other food sources for nitrate levels. And then if we have an exposed animal that's still alive, we can draw blood, test serum for that. Or if the animal's deceased, we can test ocular fluid for nitrate levels. So whenever toxicities occur, whenever forages have an greater than about 1% concentration of nitrate. All right, we're going to move on, and this is kind of timely because whether you believe it or not, depending on your location, spring is coming sooner for those of us in the southern part of the state. We've already got daffodils blooming here in Du Bois County. I might not mention that if anybody from DeKalb is on the call, because boy, it sounds like you guys had a lot of ice. But early spring related issues usually occur when animals are grazing lush forages. So we're about ready to have our early spring green up, I think, coming pretty soon. And those cool season grasses that have that rapid growth phase in the spring, that can have some diluted nutrients, high moisture content, and with that, we get some diseases that can occur. So these are diseases to be on the lookout for, I think, in the coming month or two. Grass tetany, bloat, and enterotoxemia, all of these kind of fall under the umbrella of grazing lush forages in the spring. So grass tetany, I've certainly had veterinarians tell me that they've seen grass tetany in every month of the year. So it doesn't have to be an early spring issue, but you oftentimes see it when animals are grazing rapidly growing lush forage. So those forages may have a low magnesium content, commonly occurs early in the spring. Orchard grasses, perennial ryegrass, timothy, also cereal grains have this issue. Plants that have about less than 0.2% magnesium or greater than 3% potassium and nitrogen can be affected here. So the disease that we commonly associate with this is called grass staggers. You may have heard that before. The medical term we use for it is hypomagnesemia, so it's low magnesium in the blood. And this affects all sorts of different metabolic processes within the body. The reason people call it grass staggers is sometimes animals will stagger. There is paralysis of muscles, so animals will go down. Sometimes you'll see muscle spasms, vocalizing. I have also heard people say that animals can be aggressive in this state. Oftentimes, though, most people just find these animals dead, and that's when they come to me. It can be treated if you do find animals that are affected, so you can give magnesium solutions in a variety of different routes. Again, we can test forage materials and serum in a live animal or ocular fluid in a dead animal for magnesium levels. A lot of people, if you have beef cows this time of year, are feeding a high magnesium mineral mix to try and combat this, for instance. Bloat is a disease that affects all of our ruminant species. And I think probably most people who keep ruminants are familiar with this condition, accumulation of gas within the rumen and the reticulum, which are the first two compartments of the ruminant stomach. And when we're talking about grazing, bloat comes in a couple different flavors. Of course, there's what we might term feedlot bloat, which is from high grain rations and lots of readily fermentable carbohydrates. But there's also pasture, what we call pasture bloat, and this is when forages, and these are usually forages that are high in protein, they'll produce what we call stable foam upon digestion. And so lush forage, low in fiber, maybe higher in protein, is what we worry about here. So those end up being a lot of legumes. White clover is really notorious for this, but alfalfa and winter wheat also. It's most common in the immature, the pre-bud or bloom stage of legume pastures. Usually occurs with heavy grazing or aggressive feeders, and incidence does increase after rain or dew on pasture. So animals that are kept up, for instance, and then put onto a paddock in the early morning when dew is still on, a new paddock that hasn't been grazed before, that's maybe when you should be a little bit concerned about that bloat. So clinical signs for bloat include first, left-sided swelling in the abdomen, difficulty breathing, tongue protruding. I do see a question in here. So we'll stop for a minute here and maybe try and address these questions, or maybe we might address questions at the end here. So keep putting them in the chat, and then we'll kind of talk about them because it might be some similar answers for all these. So appreciate the questions, keep them coming. The treatment for pasture bloat, this, depending on how severe it is, and there can be different levels of severity, it can be a life-threatening situation. It can be a little more chronic, a little less life-threatening, depending on severity. But in life-threatening situations, passing a stomach tube and trying to get some of that gas off is really important. If that can't be done, then a rumen trocar is what you might use. This is what a rumen trocar is, and it involves basically poking a hole in the side of the animal and relieving gas distension. This is what I look for on the autopsy floor when I'm trying to diagnose bloat. This is called a bloat line, and this is the inside of the esophagus, that tube that leads from the mouth down to the rumen. And you'll notice that there's a line right in the middle there, a line of demarcation. And this kind of reveals an interesting thing about how bloat develops and how animals actually die from bloat. So there's the bloat line, that's what we look for. And that bloat line is going to be right at the thoracic inlet, so right where the neck kind of transitions into the chest. And the reason for that is because as the rumen expands, it creates a lot of intra-abdominal pressure, which translates into intrathoracic pressure. And that pressure is so great that it actually collapses the veins that are draining into, for example, the jugular veins that are draining the neck and going back down towards the heart. So as they come into the thorax, they actually get collapsed. So all of the blood stays kind of in the head and neck outside the chest. And that's what leads to this bloat line, this characteristic bloat line. So there's the blood. That arrow is supposed to represent the blood, kind of stops because there's increased pressure. So that's one of the things we look for. And this is one of those, this kind of is to remind me too, to say that the reason that animals die with bloat is because of cardiovascular collapse. These animals just can't get enough blood to the heart and pump it out to the rest of the body. So that's why this is really a medical emergency and why it needs to be dealt with pretty quickly. This is a picture from a textbook that shows an animal that was experimentally bloated. It has a rumen fistula, so an opening to the rumen. And that lab worker here has removed the cap to that, and there's a stream of rumen contents that were under high pressure that are coming out. So this is just to demonstrate how much pressure can build up in that rumen. Clinical signs, like I said, depending on the severity, animals can be dead in a matter of hours, or it can take days depending on how severe it is. If they can get some air out of that rumen, if they can belch it up, that can prolong the clinical course. So what management strategies decrease bloat? So management systems that encourage rapid ruminal clearance or decreased rumen fermentation, mostly dealing with maybe assessing what forages are present that could be bloat prone, like those legumes, for instance. So decreasing percentage of legumes if you have a problem with bloat, for instance, assessing maturity before grazing, making sure to not graze wet pastures, for instance. So uniform, anything that improves uniform and regular intake of pasture is helpful. So patterns that also encourage more intake of dry matter, smaller paddocks maybe, and making sure that animals don't graze something down and then move to the next where it's really lush, for instance, making things more uniform. There's really no available tests for bloat, but as I mentioned, there are some things I look for on the autopsy floor, and case histories are really helpful too. Really helpful too. We talked about treatment a little bit earlier. All right, I'm not sure how many people keep small ruminants here on this talk, but we'll talk a little bit about some small ruminant related diseases, and this would be one of them. So the name of the disease is called enterotoxemia. It's an overgrowth of a bacteria called Clostridium perfringens type D. And anybody who keeps small ruminants, sheep and goats, probably have heard of the vaccine CD&T, is what people commonly refer to it as. And so this is the D in CD&T vaccines. This is what you're trying to prevent. There's a picture of the vaccine. Cattle are rarely affected by this, and usually they're cattle on feedlots. So I was really unaware this disease kind of when it's taught in the veterinary curriculum, at least when I went through, was really branded as kind of a grain associated disease. But when I started seeing this but when I guess when I became a diagnostician down here in Southern Indiana, a lot of people are grazing. When I started here, I realized that this disease does occur in animals that are on really lush pastures, and especially sheep and goats. So, like I said, usually associated with grain, but can also be associated with really lush pastures. What likely happens is a large amount of starch passes into the small intestine because there's so much coming into the rumen, it doesn't all get digested, and it passes into the small intestine, and it results in overgrowth of that kind of, if you will, bad bacteria, that Clostridium type D. So sudden changes in diet, flora can't necessarily adapt. Animals can have a couple different presentations here. They can have neurologic signs and just, have those convulsions, drooling, heavy breathing, other signs related to brain disease, or they can just be found dead, for instance, in that acute phase. Goats, interestingly, can have a more chronic form of disease in which they have diarrhea. They can have chronic diarrhea, but no central nervous system signs. And so this is kind of depending on the animals that you have, the type of animals you have, you might want to look for different things. So what we can do, we can culture intestines. We can also test for the toxin produced by the Clostridium in intestinal contents or feces. So if you have live animals and you suspect this, for instance, goats with chronic diarrhea, we may be able to test feces for this. Treatment prevention, vaccine is important. I think you need to do this every year for a few years on ewes that you're going to keep in order to consider them fully protected. I know people that boost this every year as well. Slow diet changes, reduction in intake, also important for these animals. All right, this forage-associated condition is a little bit different in that it requires some pretty specific conditions in order to be toxic. And so for those of you in Northern Indiana, this may not be as relevant of a problem right now because this forage, Dallas grass, Paspalum dilatatum, also known as bahia or crown grass, is a South American species. So it's a subtropical and tropical species that's commonly used as forage, I think, in South America. It looks all for the world like crabgrass to me. It's in these short, thick clumps and spreads via rhizomes. Mostly a problem, as you can see, that map is location, kind of incidence of this by county, mostly located in the southeastern part of the state, but we've started seeing it in Southern Indiana. Here's a comparison between crabgrass and Dallas grass. I think those long seed heads or stalks, long stalks for seed heads are a little more characteristic of the Dallas grass. What happens here is a fungus grows within the seed heads and produces a mycotoxin. And so as you can imagine, with fungal growth and with any of these ergot toxicities, you really need to have that kind of warm, humid condition in order to have that occur. And so really the danger's in the late summer, fall of the year. Animals have muscle tremors, typically starting in the head and neck and eventually affecting the limbs. So animals will be down. They also may be swaying and weak. Cattle are more affected than sheep with this, and recovery does occur if you can remove the affected forage and get animals through that initial period. So the one time I have seen this, cows and calves were affected. The pasture was grazed down pretty good, I think, and there was quite a bit of this Dallas grass in the pasture. This was down on the Ohio River, the farm was, so quite far south within our state. One of the animals was dead and sent to me, which is how we made the diagnosis, mostly based on seeds within the rumen. This is what the seeds look like. I was kind of unfamiliar with, how they looked, and sent them off to the plant diagnostic lab in West Lafayette, and they indeed came back with paspalum. And if you look inside the seed heads, those arrows are pointing to hyphae or fungal growth within those. So we did demonstrate some fungal growth within those, and based on the clinical history, I think that that's what we had at this particular farm. I bring this up not because it's common, but because it's a disease I was unfamiliar with, but I think we're going to start to see probably in the next few years as if things get warm and grasses migrate north. This is a disease that is not fatal typically. This is just something that causes usually slobbers is what we call it, or black patch disease is what it can be called in the red clover. But red clover, in again kind of those hot humid conditions, can grow a fungus that causes those black patches that you see on the underside of those leaves there. That fungus produces a toxin called slaframine, and here's a picture of a horse affected that has a lot of slobbering. It stimulates those salivary glands to produce excess salivation. So it's usually self-limiting, can last hours to days, but identifying that fungus on plants is going to be crucial, I think, for that. We also have a test that we can do. It's pretty expensive, but given that this is a self-limiting disease, usually not a lot results from it. Just something to be aware of. All right. We're going to talk, this is another disease that I've seen usually in small ruminants, but certainly can also affect cattle as well. And that's urinary stones secondary to silicate ingestion. So silica is silicone dioxide, and it's found, well, it's solubilized in the rumen, I guess, and then silicic acid is absorbed into the bloodstream of animals, and it's excreted via the kidneys. And so whenever you have one of the ways that urinary stones develop is what we call supersaturation, where lower water intake kind of allows for precipitation of some of these elements within the urine, and then you can get stone formation. So silica is present in cell walls, and as free silicic acid in all plants. All plant cell walls have these. Some have more than others, grasses and cereal grains more so than legumes. There's some semi-arid species, mostly in the western US, that tend to have a little more silica than others. And then any matured or weathered forages tend to have a little bit more of this present, I think, as a percentage of the whole as compared to younger forages. So those are the situations you need to look for. What happens in this case, though, is stones form and they can start blocking the urethra. They can't be excreted. So animals have no urine output. They're straining to urinate, maybe rapid breathing. They seem off, for instance. This happens much more commonly in males than females, and much more commonly in, for example, wethers than rams. And that's just due to the narrowing of the urethra in those particular animals. So we can diagnose this by taking stones and doing a mineral analysis. And you really probably want to do prevention rather than treatment of these guys. Just make sure water intake is high. And if you have weathered or mature forages, look out for this disease, for instance. Some people can add salt to the feed, which increases water intake. So this is the penis of a ram actually that was affected. And just so you know why small ruminants are more affected than, for example, cattle, small ruminants have this long appendage, this what's called the vermiform appendage at the end of the urethra. And there you can see that's the tan structure. I'm not sure if you guys can see my pointer or not, but this is the vermiform process. And then that bulge at the end of that is a stone. So there's the vermiform appendage removed with the stone. And as you can imagine, that stone blocks urine outflow. And animals can rupture the bladder. And ruminants, because of the way the rumen can recycle urea, rumen bacteria can recycle urea, urinary bladder rupture is not immediately fatal in ruminants. Ruminants can live for several days actually after urinary bladder rupture. All right, just wanted to acknowledge some folks, and then I'm going to switch gears a little bit and we'll visit about the other topic that Alicia had suggested, and maybe we'll take questions now kind of while we're stopped for a moment. Okay, so in the chat, we've got Dawn asked, why are nitrates high? So nitrates can become high in certain situations like drought situations. There's certain plants that are prone to do that, like corn or sorghum or Sudan grass. Johnson grass is typical too. And those plants are prone to accumulating nitrates. So certain plants just are. And they're higher usually in drought situations. They'll accumulate nitrates in those drought situations. So the makeup of the plant and then the environmental conditions are what make that nitrate be high. Why is magnesium low? I think rapidly growing lush forages just by nature don't have as much magnesium as forages in other states or other, what do I want to say, other times of their growth period. So those forages, for whatever reason, are just don't have as much magnesium. Is this a problem in nature where animals keep moving? Great question. Not sure about that. I think diagnostics in wildlife species is tough for a number of different reasons. I would say a lot of these are byproducts of the situations in which we keep animals, but we can't answer that for sure. And paddock management, I think that helps in a lot of these different diseases. Certainly having varied forages would help with some of this for sure. Varied forage species would help for sure. So Keith, do you have anything to add to those questions?
Yeah, there is something regarding the magnesium. If people fertilize with a higher rate of potassium than is necessary, then you get a lesser uptake of magnesium as a percentage of what's taken up through the roots into the plant. So we don't want to over fertilize potassium. That will reduce magnesium uptake.
Yeah, good point. Good point. Also in the chat, Don asked, Don has a comment. There's a lot more involved with grazing animals than a hobbyist would ever suspect. And I think people can have no problems, but every once in a while these problems crop up, and so just want to make people aware. Good reading that we recommend. Do you have a good a good resource for this, Keith?
Well, I think in terms of just general pasture management, there's an online publication that still, I think, has value. That's a Purdue Extension publication AY328. AY328, Management Intensive Grazing in Indiana. The other thing that's in the works that I would encourage you to look for that I think is a handy guide is the Purdue Forage Field Guide. The 4th edition is now with the editor, and I think it ought to be available to us as we get into mid-spring. It certainly should be available, and that will be through the Purdue Education store that can be ordered.
Cool. Oops. So the other topic that Alicia asked me to visit on was livestock disorders associated with preserved forages, and mostly meaning ensiled forages here. So we'll talk a little bit about fermentation basics and how ensiling works. We'll talk about a couple different diseases associated with silage, listeriosis and botulism. And then we'll talk about management recommendations for those as well. So fermentation occurs in a couple different stages. There's the aerobic stage within about the first day, a lag phase for another day, and then the fermentation phase lasts for about a couple weeks. And then there's what we call the stable phase. So in the aerobic phase, this is when all the bugs that are still there, all the microbes that are still there, and maybe the plants themselves too, are still using oxygen. And so oxygen gets used up because in the system, we packed it tight, so there's no oxygen going to get in, we hope. And so oxygen gets used up. So that blue line is oxygen and that goes down. The pH is up here at, let's say, around 7-ish, neutral, that black line. And then as oxygen gets used up, there are certain bacteria that don't need oxygen to grow. Those are called anaerobic bacteria, and they're in the red line. And they're going to start proliferating after the oxygen goes down. And so they proliferate at the same time they're producing acid. A lot of these species produce acid as a byproduct. And that acid, those acidic products, make the pH go down. And then that's what actually preserves the forage. So it pickles it, essentially. And so the pH line goes down as those bacteria come up. And then the pH, as it gets higher and higher, or well, lower and lower, the acid content gets higher and higher. A lot of those bacteria species are then killed. So the magic number with pH is to get to 4.5. You want to be below 4.5 pH to inhibit some of these problems, botulism and listeriosis, namely. We'll talk a little bit about listeriosis as a disease, and then botulism. So listeriosis is caused by bacteria, Listeria monocytogenes. This bacteria is both aerobic and anaerobic, so it can grow in air, and I think it prefers that situation, but it can grow without air. It's usually soil-borne, but boy, this thing is just about everywhere. Both of these bacteria, the one that causes botulism and listeriosis, both of these are pretty widespread throughout the environment. Listeria is interesting in that it can grow at a very wide temperature range. And so we typically see this disease in the winter time. I've seen several cases over the last couple months. I think Listeria grows fine at cold temperatures, whereas a lot of other bacteria don't. So it doesn't have a lot of competition. Because it is aerobic in some parts of its growth, or in some situations, silage surfaces are at risk, or pockets of aerobic deterioration, or loosely packed or poorly fermented material too. And also in those situations with high soil contamination. But remember, it is a facultative anaerobe, so it can grow within. It doesn't have to just grow on the surface or within pockets of aerobic deterioration. So in animals, we'll usually see 3 different manifestations: encephalitis, or inflammation in the brain; sepsis, a body-wide bacterial infection; or reproductive loss. So what does encephalitis entail? These are usually central nervous system signs: head tilt, circling, facial paralysis, animals that just don't seem with it. They may be pressing their head against walls or fences, or they may be down. And circling is such a common manifestation of listeriosis that some farmers call it circling disease. It's interesting to note in small ruminants, I've seen several cases of listeriosis in which the only complaint from the farmer was, I found these animals dead. This is a small sheep that has a head tilt, just to put some pictures in here of what we're talking about. Reproductive loss, another very common manifestation of listeriosis. So animals will abort or have stillborn animals. This usually happens late in gestation. Placental retention is common, and affected cows may have fever or anorexia. It's interesting to note that outbreaks of encephalitis and abortion usually don't occur concurrently. So a herd will either have encephalitis or abortion. We don't entirely understand why that occurs. This is a picture of a liver from an aborted fetus. This is a calf that I saw last month, and there are several, again, not sure if you can see my pointer, but there are several small white to tan foci throughout this liver. Those are areas where there's bacterial growth and cell death, secondary to listeria infection in this fetus. The third manifestation I want to talk with, and we'll spend a little bit more time on this one, is sepsis. This is one that some people don't think about with listeria. I have called it weak calf syndrome, or as Keith pointed out before, weak lamb or weak kid syndrome, also a possibility here. But these animals, they're usually a few days to a few months of age, and we think they likely become infected in utero. And I've got some cases actually where we've demonstrated that, I think, pretty nicely. So they actually get infected in utero, and then they're born live, but they're weak. They just don't do very well, and then they die, and lo and behold, we culture listeria from them. So much more vague clinical signs than what you might see in encephalitis, for instance. These animals just don't want to eat. They're kind of blah. Many people say they just aren't growing, so they're poor doers or they're failing to thrive. So in the spring of 22, we had 3 herds here that I want to highlight to kind of show you this weak calf syndrome. These are all cow herds, all beef cow herds. So first one, we got a 4-day-old calf. It was observed nursing, but it was thin. It just wasn't growing very well. Another herd, we had 2-week-old calves, so a little bit older, and these just, they said, weren't thriving like the others. And then in the third herd, I had a calf that was about a month and a half old, and that herd had had previous loss of calves that had just been born. And so in that herd, in that animal that we got, that 48-day-old animal, there were some pretty chronic lesions, bacterial infection in the kidneys that we grew listeria from. This first herd was feeding orchard grass baleage to cows. The second herd had corn silage, and this is where I think we can demonstrate pretty well that the exposure comes in utero. The cows were started on silage. They had not been exposed to silage prior to the last week of February. And then the cows started calving the first week of March. And then it was the second or third week of March, we started getting these 2-week-old calves that just weren't doing very well. And we cultured listeria from. So I think that exposure to silage in the cow, literally the last week of gestation, the last 1 or 2 weeks of gestation, allowed that listeria then to get to the calves in utero, and then they were born live, but just weren't doing very well. And then that last herd also fed silage, so a pretty good risk factors in all of these herds. Unfortunately, there aren't ways to diagnose this ante-mortem. We really need to do an autopsy of the affected animal and culture that listeria from affected tissues. There are some treatments out there. Response depends on timing or severity, I think. So oftentimes maybe the first animal is a little far along and we can diagnose it on an autopsy, and then once people know what to look for, or anybody starts to look off, if you hit them with the antibiotics, you may be able to help the next ones. All right, we're going to move on to botulism, another bacterial disease, but interestingly, it's not the bacteria itself, like listeria, that causes the disease. It's a toxin produced by the bacteria. So Clostridium botulinum, an anaerobic species, naturally found in decaying vegetation, also present in the soil, forms what we call stable neurotoxin while it's growing. Lots of different sources of risk. We talk about it here in this context mostly in relation to silage or baleage. But decaying grass, forage that gets wet, dry hay where a snake or other carrion has been incorporated into the bale can also be risks. So a lot of people think about botulism happening in carrion, for instance, but just decaying vegetation itself can also be a risk. There's a gradient of susceptibility here among species. Horses are exquisitely susceptible to botulism. Ruminant species, a little intermediate. Carnivorous and omnivorous species less so. And when veterinarians learn about botulism, horses are kind of the poster child really for botulism. And so we kind of learn about species that are really susceptible. So the clinical course is very fast. They become weak, they go down, they can have colic signs, but they're down and out and dead in 2 to 3 days. Tongue retraction is poor, so you can actually pull the tongue out. If the horse can't pull the tongue back in, it's a real sign of weakness here. In cattle, it's looked a little bit different. The cases in cattle I've seen have been more prolonged. So we're talking, instead of days, we're talking weeks, essentially, where these animals seem to be off. Oftentimes animals will go down, and the history I've gotten from people is, well, my calves just won't get up, for instance. They sit there and they look alert and they chew their cud, but they're just not getting up. Sometimes they do look stiff. Forage testing can occur in botulism. Also, contents of the GI tract we can test. The problem with botulism diagnostics right now in the US is there's only 2 places that perform the test for botulism. The National Vet Services Lab in Ames, Iowa, and then the California Health and Food Safety System. So, boy, the mouse bioassay, it's not a great test. You've got 2 mice. One of them gets botulism vaccine, the other one doesn't. You inject the material into both mice. If the one with the vaccine lives and the one without the vaccine dies, lo and behold, you've got botulism. It's only about 50% effective. It's not a very sensitive test at all. At Purdue, we're working on something a little bit better. There are a few human health public laboratories that use a method based on mass spectrometry to identify the toxin, the actual protein that causes botulism within forage material or within GI contents of affected animals. So stay tuned. Hopefully we'll have a better test than injecting the mice here really soon. Prevention and treatment, oh boy, supportive care really is about what you can do. Antiserum is cost prohibitive, really. Vaccination is available for horses, I think, but in the United States, not for ruminants. So preventing that exposure is much more critical, I think. And that's how we get to management recommendations. For both of these diseases, it's going to be very similar. So you want that target pH to be below 4.5, and you really want to avoid heavy soil contamination if you can do that. So ash content, you really want to be below 10%. Normal ash content, depending on the species of forage, is right around 5%. When it starts to creep up to 10 and over, you've got some problems there, indicative of heavy soil contamination. Both of those things kind of point us into the direction of forage testing. So if you don't know either of these things because you don't test, you can't mitigate your risk. So forage testing, I think, to include both of these are important if you're feeding ensiled forage of any kind. Don't feed obviously spoiled sections to avoid that aerobic contamination and possible listeriosis. Slow introduction of silage, or sometimes people have a lot of silage and they know that it's not great. It's got a heavy soil contamination or the pH isn't good. Maybe mixing that with dry hay is important, or mixing it with silage that does have lower ash content can help mitigate the risk. This is a view from my lab, not today. It's a sunny day outside here today in southern Indiana, but this is what the Southern Indiana Purdue Ag Center looks like with a fresh blanket of snow. Here's some contact information, and of course, Alicia has my contact information too, but you can get a hold of me by email address, gburcham@purdue.edu, and then that's the phone number for the lab. So if you have questions about how to diagnose animal disease, you can give me a call. And I think it's really important to note, I'm going to stop the screen sharing here, Keith, if I can figure out how to do that. It's important to note that there are 2 labs in the state. I know that I may be coming to you in northern Indiana as opposed to southern Indiana where I am, but in northern Indiana, the lab is located in West Lafayette on the main campus of Purdue. So there are folks there that do what I do, and including our toxicologists who can answer a lot of great questions about some of these tests. So if you've got questions, call over to the West Lafayette lab or call me. We'll try and get you in touch with someone that can help.
Are there questions for Dr. Burcham? Certainly you can chat, or don't be afraid to unmute and ask as well, please. I noticed in one of your slides, there's a couple of letters that I had to kind of play in my mind that I didn't understand. It was SQ. What does SQ in your terminology mean? It was, I think,
Oh, right. Yeah, subcutaneous. Sorry.
oh, okay, sub-Q. Yeah, okay,
Yeah,
sub-Q,
so
SQ.
right, yeah. So some people abbreviate that SC.
Yeah, yeah, yeah.
I've seen it both ways, but yeah,
Okay,
subcutaneous.
subcutaneous injection. Okay.
Yep, yep.
Very good.
Hey, this is Greg.
Hey, Greg.
Is this strictly ruminants? Like, what about birds, chickens, swine?
Yeah, so in any of it or any disease specifically?
I'm brand new to this. I've got birds. We're looking at putting some birds on pasture. Maybe some.
Oh,
chicken.
okay.
Yeah, so just wondering
interesting.
about all of this in general.
Yeah, great. So I'd say most of it was geared towards ruminants, but birds are very susceptible to botulism. So especially waterfowl. I don't know what kind of birds you have or you're going to have, but and pigs are, like I showed in that species gradient, a little less susceptible to botulism. Birds are very susceptible to botulism. So the place that birds usually get exposed is sometimes you'll hear about big die-offs with waterfowl because there will be maybe shallow water, it's late in the year, and there's a lot of decaying vegetation in the water, like a lot of decaying plant matter. And so that in that water, that botulism toxin, so the botulism is growing in that decaying plant material, that toxin will kind of diffuse out through the water body, and that can kill large groups of waterfowl, for instance. So animals sometimes in waterfowl and ducks, it's called limberneck, because the paralysis and weakness will start to affect the head and neck first, and they'll just kind of hold their head down on the ground. They can't get their head up real well before then they die. So if you hear or read about limberneck in waterfowl, that's botulism intoxication. So birds, I would say, would be, botulism is important for birds. I've also diagnosed botulism in chicken operations as well. So birds can actually harbor certain strains of botulism within their intestines. And so high fecal buildup and exposure to that can sometimes lead to botulism outbreaks in housed poultry. It sounds like you're going to be a little bit different situation because they're out on pasture, but where I've seen it in birds has been waterfowl and also housed poultry.
Excellent. Thank you.
I know we're off topic a bit, but do you have any comments about avian flu at the moment?
Yeah.
Where are we at in the state? That may be worth just a little bit of a talk about.
Yeah, sure. The ADDL, the Animal Disease Diagnostic Lab, involved pretty heavily with testing for avian influenza. Probably everybody here's heard a little bit about it. The United States and North America, really the world, are involved right now in a bird pandemic, which is avian flu. It's been going on here for about a year. It's been going on in Europe and Asia for a couple of years at this point. And it's a highly virulent strain of avian influenza that carried by waterfowl. Unusual for avian influenza, it's actually causing death in waterfowl as well. And when that crosses over into poultry, it can cause massive death loss. And so we have sporadic cases still occurring. Not in Indiana. There is a case recently diagnosed in Southern Illinois by a company, actually an Indiana company that has birds in Illinois. So it's cropping up here and there, something to keep your eyes on. If you do have backyard birds, worth really keeping an eye on them. And if there's any unusual mortality, getting them tested, I think, for avian influenza.
You've got some chat there, Grant.
Oh, thanks. Yep. Yeah, when animals are diseased, can any of these be zoonotic to humans? Listeria certainly is a disease that affects people as well, but not necessarily contagious from animals to people unless you're eating animal products, for instance. So certainly that's not going to be going into the food chain because we don't slaughter, sick animals for food. But I guess the same sort of exposures might exist. People aren't eating forage, for instance, but where it usually happens in people are, certain food products that are prone to growing it, like lunch meat, for instance, that's in the fridge for a long amount of time. That's why pregnant women shouldn't eat lunch meat. That's the recommendation currently from public health officials. So there have been a lot of Listeria outbreaks in lettuce and I think ice cream recently. So it crops up here and there in various different food sources. Can affect people. Not being around livestock is not a risk factor really for listeriosis. It's mostly foodborne for people. Any of the other diseases, I would say not in this particular instance are zoonotic, but always something to keep an eye out for. Avian flu, there have been I think a very few number of instances of poultry workers affected with this strain of avian flu has resulted in mild illness so far. What labs that do forage testing, test for botulism using the mouse bioassay procedures? Oh, that's interesting. I'm not really familiar with the forage testing scene, Keith. Maybe you would be better equipped to answer that. Does anybody test for botulism in the forage kind of testing world?
Not that I'm familiar with. No,
Okay.
they do not. Yeah, do not. I don't know of anybody that's housing mice to do the test in a forage testing facility.
Yeah, good question.
The greatest key on that is to get those forages analyzed and look for that pH of 4.5 that Grant mentioned.
And the ash content too. That's something that.
And the ash content, absolutely. We have seen that time and time again from, I think, a couple of things. First of all, over tedding. Some of our forages, in particular, I think about sorghum Sudan grass or Sudan grass. It's high moisture content, and we try to get that in a wrap bale, and to help that get to that 50% where we should be wrapping, we can get a lot of ash. The other one would be that I've noted is small grains like rye. I think you've seen some rye problems as baleage. You all probably, if you've been around the farm, can envision that tractors getting out on a soil that's a bit too wet really to be there, but they bring up, if you will, wet soil clumps that we would probably, as they dry, call clods. And that can be easily gotten into a windrow and then baled up, and that would be a source of Clostridium bacteria as well.
Yeah, I know at least one of the cases of botulism we had when they looked at ash content, it was about 14%.
That's double the amount that you would expect or more. Yeah.
So once you get above that 10%, I think you're looking at possible issues.
Yeah. Any other questions? Here we go. We got another one, bud.
Yep. I've always heard that clover causes a horse to slobber. Is it only the diseased clover that is an issue, not healthy clover? Great question for Keith. I don't think non-diseased clover would cause that slobbering, would it?
Absolutely, you are correct. Really interesting case. There was a place, David, I think I know about where you live. It was in a region of the state, and I actually visited this particular farm that was really interesting. Low-lying area around the stable. Dew did not come off in that particular area, typically in the summer until mid to late morning, and it was just the right environment for the Rhizoctonia leguminicola that produces the fungus that produces this slaframine to have that happen. So as with all diseases related to plants, and I'm sure it's true with livestock too, Grant, environment's a big part of this as to whether it expresses itself. So you can have the pathogen potentially present, and you got the host plant, but if you don't have the right weather conditions, it may not produce the bacteria. So you are correct that clover that's growing properly without the disease symptoms in it, literally, the leaves on red clover just look like black, graying, dying leaves is what black patch disease looks like.
Yeah, good question.
Yep. Oh, the other thing about that's really interesting, as compared to a lot of things that don't go away in time, research has shown that slaframine store red clover hay for a year, and you'll find that content does decline, which is a bit unusual in a lot of toxins.
Yeah, that's true. A lot of these, well, we didn't talk anything about toxic plants, but in a lot of toxic plants, usually the principle is still there. Toxic principle is still there after drying in hay.
Any others? Well, as I will always note, it's great to do these forums and these webinars, but we give our information to contact for a purpose. And so make sure you reach out to Dr. Burcham if you have questions as it relates to his expertise, and myself as well. And certainly I may not be able to answer the question, but I do know of people in terms of a network that could. So do we have? Yeah, go ahead. Grant, looks like one last one. We'll call it a day.
Okay, that sounds good. And this is probably a topic for another day. It says, can we talk basic animal nutrition from forage? That might be a really good topic, I think, for your forage forum here. Is nitrite equivalent to what we call protein in human nutrition? No, is the answer there. And then is there a carbohydrate requirement? Certainly and I think carbohydrates are usually measured in energy, right? Is how
Correct.
people like forage tests will show an energy number, and that's basically the carbohydrate number. So you get some forage testing done for basic nutritional analysis, you're going to have protein, energy, probably fiber percentage too, right?
And minerals.
And minerals, yeah. So so you can measure protein, and that's separate really from nitrates and nitrites.
The thing about the carbohydrate question, you've got non-structural carbohydrates, which would be the simple sugars, glucose, fructose, galactose, those sorts of things. And then you've got the structural carbohydrates, which would be like cellulose and hemicellulose. And that's what, of course, a ruminant or a horse can use because of the microflora of the rumen or the cecum. Yeah. Well, thank you, everybody. I thought we had a great experience, and we do have a forage forum Friday next week as well. I believe this will be Dr. Steckler from the University of Illinois, if my memory is correct. So we encourage you to show up again and invite others to attend. Also know that these presentations are stored, and I'll have Alicia make sure she gets out to where the YouTubes are stored of the past year and a half or so that we have. There's still good up-to-date information, but feel free to share this with others to let them know that we do have information here related to forages and lots of different attributes. But I'd like to thank Dr. Grant Burcham for being our expert today. A very fine diagnostician, and thank you very much, Dr. Burcham.
Yep, happy to be here. Thanks, everyone.
Take care, everybody. Be safe.